All right, on that last part, I was finishing up with osmosis, or was covering osmosis. So all osmosis is the diffusion of water across the plasma membrane. Same process as diffusion, except it's for water. Okay, so these green spheres would be water molecules diffusing down their concentration gradient across the plasma membrane. And with facilitated diffusion, I'll show you the figure for that in a minute here, or second here, a few seconds. That is the process of the diffusion of a substance or solute down its concentration gradient across the plasma membrane, except now there is a transmembrane integral protein that allows the diffusion to occur, basically facilitating diffusion. So I'll go back to this figure here. So this integral protein that spans the membrane acts as a channel that would allow facilitated diffusion of some substance or solute across the plasma membrane, whether into or out of the cell. Okay? So the force that's moving the material across the plasma membrane with diffusion, osmosis, and facilitated diffusion is simply the concentration gradient. Whenever there's a concentration gradient, diffusion will occur, unless it's something that cannot get across the plasma membrane on its own. So here in figure 2.4, discussing diffusion. In this case, carbon dioxide diffusing out of a cell across the plasma membrane, and then the carbon dioxide would diffuse into a blood vessel, and then your cardiovascular system transports the carbon dioxide to your lungs where you breathe it out. Okay? So read the information here in figure 2.4 so that you get a good grasp of diffusion. And there's osmosis, same process in the sense of water is moving down its concentration gradient. In this case, the water is moving out of the cell down its concentration gradient. And then here's facilitated diffusion. Some substance like blood sugar, glucose, enters into your cells with the assistance of an integral protein, which is a carrier protein specifically for glucose. So there's no energy required to have the glucose enter into the cell. What's pushing the glucose into the cell is the concentration gradient of glucose. So glucose, blood sugar, enters into your cell via facilitated diffusion. So continuing with membrane permeability, let's look at active processes. Processes that would move a substance, in this case this red molecule, across the plasma membrane, but energy is required in the form of ATP. So one process is called active transport. So this membrane protein in purple is able to latch on to some substance and move it across the plasma membrane, but it's using ATP energy to do this. So this is an active process, active transport. Endocytosis, notice the word roots, endo, inside of or internal, cytosis, cyte, the cell. So endocytosis is the movement via active transport of some substance into the cell. So in this example, the top would be internally, inside the cell cytoplasm, and down here at the bottom would be external, the extracellular fluid or matrix. Two types of endocytosis is pinocytosis and phagocytosis. Pinocytosis is sometimes called cell drinking. So that's the process of moving relatively small substances into the cell. Phagocytosis we'll look at a little more closely, and that's where large substances or whole cells like bacterial cells are moved into the cell. Both processes require energy. They are both active transport processes. Exocytosis, exo meaning out of or external, is the opposite of endocytosis. Exocytosis is moving a substance out of the cell through active transport. So if this was exocytosis, down here at the bottom would be inside the cell. The particular substance is moved and exported out of the cell with exocytosis. So here's active transport, figure 2.4. Right? This example is showing what's called co-transport, but the movement of something inside the cell in addition to moving something outside the cell. So this example is called the sodium-potassium pump. Potassium Ka+ is being moved into the cell, while sodium Na+ is being moved out of the cell. And when we look at the nervous system and the generation of nerve impulses, we'll see that the sodium-potassium pump is important for maintaining the correct concentration of sodium outside the cell, extracellular fluid, and potassium inside the cell within the cytoplasm. But here's the ATP energy that's required for this pump to work. ATP energy, the ATP latches onto this pump, which is an integral protein, but it's called a pump because it's pumping material into and or out of the cell. It will not operate without the input of this energy in the form of ATP. So ATP provides the energy. When ATP releases its energy, it forms ADP with the loose phosphate, which is not shown here. And the energy provided by ATP is used to pump, in this example, potassium into the cell and sodium out of the cell. Pinocytosis, movement of small materials into the cell with the requirement of energy input. That's not shown here, like the ATP molecules aren't shown, but it is required that energy input be utilized because it is an active process, active transport. And then here's phagocytosis, literally cell eating. Phago refers to ingestion or eating, and then cyte means cell. So like your white blood cells will phagocytize something like a bacterial cell, and in this case it's not just a small portion of the plasma membrane that forms what's called a vesicle, a little bubble in a way, to import something. It's extending extensions of the plasma membrane called pseudopodia. Pseudopodium is one of them. The pseudopodia extend, engulfing the cell in this case, forming a large vesicle, phagocytic vesicle, that gets moved into the cell, and then that item is destroyed inside the cell through the action of digestive enzymes. So two active transport mechanisms of endocytosis, pinocytosis and phagocytosis. Okay, exocytosis, the movement of material outside the cell. It's basically the opposite of pinocytosis. Instead of something being moved into the cell, it's being moved out of the cell. So some cells produce proteins and those sorts of other sorts of molecules for export. The cell will produce protein, or proteins in this case, package it in a transport vesicle. The vesicle moves to the plasma membrane, fuses with the plasma membrane like two soap bubbles fusing, and then what was inside the vesicle then gets ejected outside of the vesicle. Exocytosis, its active transport requires ATP. Now let's look at extensions of the plasma membrane called microvilli. So these are finger-like projections, extensions of the plasma membrane, and they primarily function in increasing the surface area of the plasma membrane to increase the rate of absorption or secretion of materials across the plasma membrane. So increasing SA, so increasing surface area of the plasma membrane, that increases the absorption capacity from the extracellular fluid out here. So the cells that line your intestinal line, your small intestines and your kidneys have ample, many microvilli to increase the ability of those cells to effectively absorb and move substances across the plasma membrane into the cell and in the small intestine to get those substances then into your bloodstream. Okay, so microvilli, extensions of the plasma membrane, increase surface area, increase absorption ability, or even secretion ability of particular cells. The cytoplasm, all of the material inside the cell, excluding the nucleus. So the cytosol is the fluid-like environment inside the cell. Consists of what's called the intracellular fluid, mostly water, lots of dissolved nutrients like glucose and proteins and waste products within the cell. And then the organelles are the specific structures inside the cell that have specific functions. Okay? So the cytosol contains higher, in general, concentrations of potassium and lower concentrations of sodium versus the extracellular fluid. Go back and look at that sodium-potassium pump that helps maintain these concentration differences. So because of these concentration differences, there's a resting membrane potential where there's a relatively negative intracellular charge just inside the plasma membrane compared to just outside the plasma membrane, which has a relatively positive charge. So a positive extracellular charge. And this is maintained in your cells, and that's called usually the resting membrane potential. And that can get altered and changed when an action potential or nerve impulse is generated. So we will revisit this when we talk about the nervous system. And also within the cytosol, there are dissolved proteins, carbohydrates like glucose, amino acids, lipids, whatever substances the cells need to be healthy, basically. There's also insoluble substances, things that basically kind of clump together, if you will, things like glycogen. So in your cells, if you have abundance, an excess of glucose, which are small, simple sugars, they can be linked together to form large, complex carbohydrates, storage energy molecules like glycogen. In particular, in your muscle and liver cells store ample amounts of glycogen under normal conditions. And these type of insoluble substances that do not dissolve easily in liquid water are called inclusions. A comment here. Sometimes students will get confused about cytoplasm and cytosol. So cytoplasm is everything inside the plasma membrane, usually excluding the nucleus, which we don't see here in this diagram. And the cytosol is the liquid material of the cytoplasm. And the cytosol, the liquid material, is separate from the organelles. For us, however, we can kind of use these terms interchangeably, but technically they are different. So the organelles, structures that have specific functions, as we'll see. There are non-membranous and membranous organelles. The membranous organelles have a phospholipid membrane. The non-membranous organelles obviously do not have one. They're like naked, if you will. So here's non-membranous organelles, the microvilli, because they have a external covering of plasma membrane, but not totally surrounding the entire structure. Free ribosomes, where proteins are produced, do not have membranes. The centrioles of the centrosome does not have a plasma membrane. Sorry, not a plasma membrane, but does not have a membrane, okay? There's only one plasma membrane on the outside. Excuse me. The free ribosomes do not have membranes. What else do we have here? Fixed ribosomes on what's called the rough ER do not have membranes. And then the cytoskeleton, the internal structural components of the cells, also do not have a membrane. Okay? Many of these other structures organelles do, the membranous organelles. Secretory vesicles that fuse to the surface to eject materials, mitochondria, Golgi apparatus, peroxisomes, lysosomes. The nucleus has a double membrane, actually. The rough endoplasmic reticulum, the smooth endoplasmic reticulum, and I believe I mentioned the mitochondria. Those are organelles that all have membranes. Again, primarily phospholipids, just like the plasma membrane is primarily phospholipid. So, non-membranous organelles, the cytoskeleton. Just like with your body, the cytoskeleton allows your body to move and gives you structure and support and protection. So these are protein filaments, proteinaceous filaments and microtubules. So structures that have different microanatomy, and they have different functions in the cells, but overall they provide the cells with